Okay. So for your exam two, same general format, multiple choice, true, false. Everything that I just said is on the Scantron. You have matching, you have fill in the blank, you have a labeling one just like you did before, and then short answer. So the entire test is worth 100 points. Um, you'll need a Scantron, obviously, no calculator this time, and a pencil that works for it. So feel free to start asking questions. If it's exactly the same question as someone else had asked before, and I've already answered it in detail, I'll probably just refer you to the YouTube video that is going to be coming out pretty quickly after we're done with this. So we'll go into 11. Feel free to start asking. I always appreciate when people actually ask it with a mic rather than through text, but I will take either. Dun, 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 dun.
Do we need to know the gel formation for a soluble and insoluble fiber?
Like that soluble fiber does create gels. Yeah, you should know that.
Yeah. And the gut transfer times?
Yeah, I wouldn't focus so much. Well, yeah, I would know the general differences. Like I would understand the profiles between soluble and insoluble fiber, but I would also understand that as a whole, fiber fiber intake, a higher fiber intake to a certain degree is going to speed up gut transit time and increase bulk in stools as like a whole. But within that, I would understand the differences between soluble and insoluble fiber. You should know the requirements too. All right, so the 14 grams per 1,000 calories slash 2,000 grams per 28, or sorry, 28 grams per 2,000 calories. There's the other one as well that was mentioned, the AI for it, which off the top of my head, I'm going to say is 37 grams for males and 25 grams for females, but I might be off by a couple of grams there. Yeah, Jessica asked, can we go over the transporters for glucose, galactose, and fructose? So transporters would be trying to pull individual basic unit monosaccharides from the small intestine into the enterocyte. And so with the individual monosaccharides, glucose, galactose, and fructose, we have two different transporters that bring these three in. So we have GLUT5, which is going to be bringing in fructose, and that is going to be facilitated transport or facilitated diffusion. So it does depend on a concentration gradient. And then for glucose and galactose, we have SGLT1. That is an active transporter, which means that it does not rely on a concentration gradient and does require ATP. So once all three of those are in the cell, they're going to be brought out into the blood through GLUT2. So all three are actually going to be exiting the same way. And GLUT2, just like all glutes, are going to be facilitated to rely on a concentration gradient. Technically, if I'm saying that it's facilitated diffusion, it is passive by nature, right? So active is going to be requiring ATP. If you might get confused if I say passive transport versus facilitated transport, but both are actually passive by nature. It's just that facilitated diffusion is passive but requires a transporter protein. Um, and so just understand that after the enterocyte, we go into the blood, but it's a specific vessel called the hepatic portal vein that leads to the liver and then for processing and then bringing what is necessary out of there into the systemic circulation. I guess while I'm on it, you should know your carbohydrate digestion starts in the mouth, salivary amylase. Nothing happens in the stomach, primarily because the salivary amylase is an enzyme, and enzymes are proteins, which now you'll know are denatured. by the stomach acid, so they no longer function the same way. Majority for all three macronutrients happens in the small intestine. You should also know the individual combinations for the monosaccharides into the disaccharides. Glucose makes sucrose, or I'm sorry, glucose makes maltose, glucose fructose makes sucrose, and then glucose galactose makes lactose.
What's the function of glucagon again?
It's the opposite of insulin. So I always think since people know insulin a little bit better, it's a good contrast. Insulin functions to lower elevated blood sugar, and it does that by binding to receptors on the outside of the cell, opens up a door and transport protein to allow glucose to go from the blood into the cell itself. So the overall stimulus is elevated blood sugar. The response is to release insulin, and the overall effect is to bring glucose levels hopefully back down to normal ranges. Glucagon is the exact opposite. So it's going to happen under low blood sugar instances. The way that it elevates blood sugar is a little bit different. So it's going to actually stimulate the liver to break down glycogen, the stored form of glucose in the liver. So breaking down liver glycogen, and then that glucose that's liberated from that glycogen will then be released into the blood, and the overall effect is to increase blood glucose. Both glucagon and insulin are made by the pancreas. It's just different cells. Insulin's made by the beta cells. Glucagon is made by the alpha cells.
So the glucagon is low blood sugar stimulates the liver to break down liver glycogen. Then
Mhm.
the glucose from that is what puts like the glucose from that then goes into your blood.
Yes, because the overall effect that we're wanting is to increase blood glucose when it was low. That was the stimulus per se.
Okay, that makes sense. Thank you.
Yep.
Professor, could you kindly explain conditionally essential amino acids, please?
Sure.
Thank you so much.
So you have essential and non-essential amino acids. So
Mhm.
just to clarify, amino acids are the individual basic units of proteins. So multiple amino acids make up proteins. There's 20 amino acids in total. Some of them are, 11 of them are non-essential, which means that we make them, so we don't have to consume them. And 9 of them are what we call essential, which means that we have to consume them, which was a term that we learned in the first unit. Conditionally essential amino acids are amino acids that are normally non-essential, so we normally make them, but under very specific circumstances, we may need non-essential amino acids the same way that we would need essential amino acids. So it basically is a non-essential amino acid that becomes essential due to a specific condition or scenario. An example of that would be if someone is an aerobic athlete, so they do endurance running or they have some form of cardiovascular disease that would be limiting their ability to properly deliver blood flow or efficiently deliver blood flow. Arginine can become conditionally essential because it plays a role in the creation of nitric oxide, which overall expands the blood vessels. And when you expand a blood vessel, it relieves a lot of the pressure, or I guess the strain on the heart to push harder because it increases the amount of fluid that can flow through that particular artery. So, did that clarify things?
Yes, thank you so much. I appreciate it.
Of course.
Did you say an endurance runner or somebody with cardiovascular disease, or did you say that they were the same person?
Oh, I mean, if they were the same person, it would be very much so conditionally essential. But it would be and or. So if you're an aerobic athlete, obviously one of your limiting factors is how well you can deliver blood flow or deliver the things that are in your blood. So blood flow is a major determinant. And then if you have cardiovascular disease, there's impaired blood flow to some degree due to all these different factors. Inflammation injuring blood vessels, not having, like sufficient vasodilation that's occurring. Um, I mean, you can get into other things like left ventricular hypertrophy and the stress that that causes. But ultimately, like if you have a higher need for your heart to function better, then you're going to have a higher requirement for something like arginine to help with the creation of nitric oxide to expand the blood vessels. Does that kind of make sense?
Yes.
Cool.
For the protein RDA, it's not optimal because I think you said last class it was because it wasn't a way there wasn't a way to measure it, right? Something like that.
Um, Not so much. So the RDA is 0.8 grams per kilogram, and that's just enough to prevent deficiency. So we talked about kwashiorkor, which is a pure protein deficiency. And we talked about marasmus, which was a protein deficiency, but it also is just a deficiency in everything else. So it's kind of like akin to starvation in a lot of ways. But the point is that the RDA is not what's optimal. It's just what is enough to prevent people from getting sick because of it. There's a difference between what's enough versus what's optimal, right? If you know that you can go throughout your day on a minimum of 5 hours of sleep per night, that's great. But you know what's optimal is to get the 7 to 9 hours. So you just need to be able to compare and contrast what is enough and what is better, right? I guess you're like your grades are a good example. So like some people will settle for like a C, and other people what's optimal is actually just to get an A, right? So um what's good enough is not always what's best. Oh, and I guess what you were referring to, Nazip, is that there's no consensus on what optimal is. A lot of different conversation about that still.
Got it.
Can we go over the neutralistic fallacy?
Naturalistic fallacy. Yeah.
What did I say? Neutralistic? Naturalistic. That's what I meant.
Yeah. The only reason I corrected you is because the word natural is an important component to it. So the naturalistic fallacy just states that anything that's natural is inherently better for you, which is not the case. I think that's literally, well, the naturalistic fallacy is probably going to be viewed a little bit differently now, in the current political climate that we're in. But that's a little bit beside the point. So the naturalistic fallacy, as I mentioned, is the idea that just because something comes from nature or is inherently more natural, that it's more healthy for human physiology. So a really good example would actually be pesticides, and most people think that pesticides are inherently bad. But if you look at the alternative, pesticides were originally brought into agricultural practice to prevent, as the name would suggest, pests. And so, yes, we have concerns, and obviously, I'm not saying that you should just welcome all in pesticides in any amounts, but generally speaking, the act of using pesticides as a whole has prevented pests from transferring diseases into our food stock, and allowed us to also keep crops and other um, components of food for other purposes in the industry available in large amounts over long quantities of time. Literally, like you could say everything that I just said because I'm going to ask you what the naturalistic fallacy is and what an example would be. If you really can't remember what I just said, you could say something like earthquakes are natural and they kill people all the time, right? Um, Glasses, I wear glass. I would be blind without them. But if like I think that we've been able to extend a lot of our quality of life due to things that have been the products of human ingenuity, so to speak. So yeah, you should just know that what the definition of the naturalistic fallacy is, and then the word fallacy would denote that it is false um, thing, right? So it's naturalistic fallacy. It means that the idea that just because something is natural is false, hence fallacy.
Okay, thank you.
Mm-hmm. By the way, from your video analysis one, you probably don't remember this, but the actual leptin study that was referenced, the one that had uh, the Biggest Loser stuff, that study was ran by a researcher named Kevin Hall. And Kevin Hall actually just resigned from the NIH recently due to censorship of his research under the current administration. So this is it's not a good time for science right now. I'm just saying. Anyway, continue asking. I'm just like distraught. Yeah, Jessica asked Pepsin and its role in, I'm assuming you mean HCl, which is hydrochloric acid, versus HC1. HCl is hydrochloric acid, which is the acid that we release in the stomach. HCl also plays a role in protein digestion because the acid is one of the ways that we're able to denature proteins, which makes them easier for us to be able to actually break down into their individual amino acid constituents. So the acid is going to help unfold the protein, and then the pepsin specifically is the enzyme that's going to be breaking it down. So it's going to break down longer protein chains into smaller ones, or in some instances, going to be breaking down smaller protein chains into individual amino acids. And the other thing that I didn't mention that you will also need to know more specifically as a short answer question is that pepsinogen, which is the inactive form of the enzyme pepsin, is actually activated by hydrochloric acid. So the role is that normally when we release this inactive form of pepsin called pepsinogen, it doesn't actually have the effect to break down proteins until it meets up with hydrochloric acid, which also happens to be if you just ate something where all the food is, kind of like gravitating towards the middle of the stomach. Whereas if we did just release pepsin without it being in an inactive form, it would digest the walls of the stomach because they're also made out of protein. So I'm assuming that's what you meant, right, Jessica? HCl. Cool. So hopefully that answered the question, gave you a little bit more. I mean, we got 30 more minutes, so I guess I could talk about some of the other short answer questions. No one's talked about omega-3s, omega-6s yet and the different fatty acids. So you should know that the essential fatty acids are omega-6s and omega-3s. I would also be very familiar, at least with the common acronyms for omega-3 and omega-6 fatty acids. So omega-6 fatty acids are related to pro-inflammation, causing inflammation. We get those in pretty high quantities. Those are the ones that we don't necessarily recommend to people too often because they already get enough of them just from eating a typical Western diet. And the kind of like starting, so to speak, omega-6 fatty acid is linoleic acid, which can be abbreviated as LA. That in itself doesn't play a role, but once it is converted into arachidonic acid, which you can abbreviate as ARA, that actually gets used to create the pro-inflammatory factors. Um, on the other end of the spectrum, we have omega-3s, which is commonly the one that we recommend because they're much more difficult to get in a typical diet. So those are anti-inflammatory by nature. The starting fatty acid is alpha-linolenic acid, which we can abbreviate as ALA. That is similar to linoleic acid, doesn't play a role in itself. It needs to be metabolized into first EPA, which primarily plays the role in anti-inflammation, and then a couple more conversions, and we actually get DHA, which we know very well, and you're going to have to know the primary notable function is relating to pro brain and eye health. And this is especially important for developing fetuses as well as for lactating and pregnant mothers to be able to transfer some of those fatty acids over, which makes sense though too if you keep in mind that the brain is primarily made out of fat. And getting in enough omega-3 fatty acids helps to kind of facilitate the structure of the brain as it's developing. No one asked, but the question about whether or not why salmon is actually a better source of omega-3s over something like flax or chia. So that one, plant sources typically contain ALA, which you'll remember is the form of omega-3s that we don't really want. It's the form that it has to be converted. But when we eat sources of ALA from plants like chia seeds and flax seeds, our conversion rate from ALA into EPA and DHA is absolutely horrible. So if we look at omega-3s as a blanket term, we can have as much ALA as we want, but the problem is that the conversion rate sucks. So it's actually much more efficient to consume fatty fish like salmon or tuna because they have already done that conversion of ALA into the particular fatty acids that we want, EPA for anti-inflammation and DHA for brain and eye health. So I guess the point is that even though if we were to measure total omega-3s, that they would actually be lower in something like fish, it doesn't matter because the types that we want are the EPA and the DHA, and not just looking at omega-3s as a whole.
Professor, I have a good question.
Sure.
Could you please tell us why do we need a balance between the ratio of omega-3 and omega-6?
Yeah. So the reason is kind of twofold. So the first reason is that we need a balance of inflammation and anti-inflammation. Inflammation is not inherently good or bad. Inflammation is something that we require in order to adapt and have a functioning immune system, along with many other things. And so inflammation is important. It's just that we don't want too much of it. And by the same card, we don't want too much anti-inflammation. Otherwise, we wouldn't have a proper adaptive system, so to speak. The reason that we need to get them in as close to a one-to-one ratio as we can is because the conversion from LA into ARA and from ALA into EPA and DHA. So basically, these forms of omega-6s and omega-3s that kind of are our starters that don't actually play any roles in themselves, that conversion on either side requires enzymes. And those enzymes work the same. So they are the same enzymes regardless of whether it's an omega-3 or an omega-6. So the way that I like to think of it is it's first come, first serve for access to those enzymes. If you're eating an overabundance of omega-6s, you're going to have fewer available to metabolize and actually utilize the omega-3s. So they compete with one another is how I would say that in a very brief form.
Okay. So like the enzyme will work for us, like let's say if we consume omega-6 or like if you consume omega-3, so they kind of compete each other
They
for
compete
the
with each other.
enzymes. So if this question was on the paper, um, would you want us to like describe that whole scenario, or like we just say because they both require the same enzymes, they compete for each other, therefore we need to balance the ratio, and the ratio is 1:1?
As close to one to one as possible. Yeah, I think the way
Okay.
you described it is good, and I just want to make sure that I'm emphasizing that you know why we need a ratio. So the
Okay.
competition, but also they both play very important roles in physiology because we need inflammation and we need anti-inflammation.
Got it. Thank you so much.
Yeah, of course. The last short. Oh, go for it.
Oh, sorry. For the labeling questions, I remember in class it was the protein structure. Is that going to be the labeling one on the test?
Amino acid structure, yes.
Okay, cool.
Amino group, um, carboxylic acid group, and R group.
Okay, perfect.
Yeah. And I was just going to talk about the last short answer question and as much as I can remember. I'll check after this to see if that actually is the last one. But the whole is fruit sugar something that people should be concerned with? And you should say no. The main things that I'm looking for are that the fiber and the water essentially dilute, so to speak, or reduce the ability of a particular food to increase blood sugar. So a lot of people are concerned with fruit because of the whole push towards keto. And realistically speaking, fruit isn't going to be doing anything in the quantities that we normally eat it because fruit is kind of in a package that already makes us want to eat it in lower proportions. So, like a banana, most people aren't hammering down three bananas in one sitting unless they're just complete super, super hungry. They're usually happy with just, one, maybe two. And part of the reason for that is that, yeah, it takes longer to eat because you have to do more in order to actually consume it, chew it more, right? It has fiber in it, and then there's water inside of it. And so that water is going to fill you up, but it's also going to literally dilute the amount of carbohydrate that's going to go from the stomach into the small intestine. I'm going to check to make sure that was the last one while y'all come up with questions.
So, Professor, for the fruit question, we can just say no because the water and fiber control the glucose levels. Is
Yeah,
that good enough?
pretty good. Yeah.
Okay, thank you so much.
Yeah, so I've covered all of the short answer questions. For the fill in the blank, you're going to want to know the difference between a complete versus an incomplete protein. Incomplete proteins are typically plant sources. There are exceptions. Complete proteins are typically animal sources. And a complete protein is really talking about its profile or what the amino acids, what specific amino acids are in meaningful quantities. So most animal proteins contain all nine essential amino acids in proportions or in portion sizes that would be practical for people to consume to get those amino acids. Whereas plant sources are typically incomplete, meaning that they don't have a meaningful amount of all of the essential amino acids. But what you can do, which is another term you'll need to know, is if you combine two incomplete proteins that have kind of opposite profiles, you can create something called a complementary protein, which is essentially two or more incomplete proteins that form what would otherwise look like a complete protein. More questions?
For fat digestion, it's not in the mouth. It's then goes, is it the majority of it is in the small intestine?
Everything's always the majority in the small intestine.
Okay. And then from there, that's when it goes into the lymphatic system.
After the small intestine, yeah, there's a lot with fat digestion. So when you said it doesn't happen in the mouth, just make sure that's in the context of a healthy adult. Because I did mention to you that for this exam, the way that I was taught was that for babies specifically, there's relevant digestion in the mouth because of an enzyme called lingual lipase. Um, that's mostly just because babies don't have a very well developed gastrointestinal system. And so there's a little bit more dependence on getting nutrients from like fats that are broken down a little bit earlier in the mouth. The stomach is where the majority of it happens. That happens with gastric lipase. And then you also want to be aware of the kind of unique transport of fats once it gets into spaces like the small intestine, because you need phospholipids from bile that are going to be, I guess, for lack of a better term, encasing the fatty acids, along with like the cholesterol and the fat soluble vitamins that you consume. And that's going to allow what would normally be like a hydrophobic substance. So fats don't mix with water. That's what hydrophobic means. It's going to allow them to get transported, and it's going to break up big globs of fat into small ones to be able to increase the ability for fat to kind of be digested faster. And so once those little vehicles of fats that are wrapped up into phospholipids, and those are called micelles, kind of dump or drop off their individual fatty acids into the enterocytes or the small intestinal cells. They get repackaged into triglycerides, which is kind of the backbone with the three fatty acids on it. And those get brought once again into like a little vehicle that's made up of those phospholipids, and that's called a chylomicron. That chylomicron, as you mentioned, goes into the lymphatic system, and then it gets dumped out into systemic circulation or the blood after that.
Okay. That was a lot.
Yes.
Okay. I feel like I'm starting to write all that down. I was like, I should have that in my notes somewhere.
Yeah, well, the great thing is that when you're on YouTube, you can actually slow down the video. There's like a function in the little over by where you can change like the resolution from 360 to 480. You can actually slow me down to like 0.75 or something, which is interesting because like I always wish that I could like talk and think faster. So normally people would speed me up, but I know that was a lot right there. And if you want me to specifically break that down in any way again, in like any of the individual pieces, let me know.
Okay, so because I have in my notes, fat digestion starts in the stomach with gastric lipase,
Hmm.
and then enzymes come from the pancreas to digest things in the small intestine,
Hmm.
and then bile emulsifies fat so it can be digested.
Hmm.
And then the chylomicron made from the fat we consume and I don't know, I have VLDL transports what our body makes.
So that brings up the lipoproteins. So there's four that you need to know. Chylomicrons are transporting fats and cholesterol technically from the foods that we ate. So that's that whole transport that we just mentioned in the digestive process. after the chylomicrons go through the lymph and they go out into the circulation, they're going to be dropping off all the stuff that we ate. VLDLs have a very similar purpose, but the VLDLs are actually made in the liver, so they're not what we ate. They're fats that we're producing. And then that as VLDLs go around the body, they're going to be dropping off their individual triglycerides or their individual fats. And then the VLDL, because it's dropping stuff off, is going to shrink. As it shrinks, it becomes more dense. It becomes more dense with specifically cholesterol because it was dropping off fats, not cholesterol as much. And so you end up going from a very low density lipoprotein, so big and fluffy, and it gets more dense as it drops off the triglyceride. So we call it an LDL or just a low density lipoprotein. So it goes from very low density just to low density. So that's the contrast between a chylomicron, which is from the diet, and VLDL, which is made in the liver. And then from VLDL, we go to LDL, carrying mostly cholesterol, and it's carrying cholesterol in the direction of the liver out to the body, right? Because that was the idea with VLDLs. They were starting in the liver, dropping things off throughout the body in circulation. You want to contrast LDL with HDL, which is also carrying mostly cholesterol, but it's carrying it from the body back to the liver. So you can view that kind of like traffic. You want cholesterol going out in the LDL package because cholesterol plays a structural role in a lot of things like cell membranes, different hormones, etc. But we don't want too much out there because of the risk for cardiovascular disease. By the same card, we also want HDL to be going back to prevent there from being too much cholesterol that's kind of floating out in the bloodstream inside of those little LDL packages. That was a lot too. So was that okay?
Yeah, so LDL from the liver to the body, and then HDL is from the body back to the liver.
Correct.
Okay, and then from VLDL to LDL, it's the fluffy, then condensed.
Yeah.
Okay, got
Good.
it.
About like 10 more minutes. More questions. More questions.
Hello, Professor. Could you go over the transporters for glucose, galactose, and fructose?
Sure. I actually already covered that in a little bit earlier. So
Okay.
I'm gonna put the video up. It was pretty early on as well. So
Okay.
yeah, I'm going to say it really quickly again.
That's
For glucose and galactose, it's SGLT1, which is active transport. That's to go
Mhm.
into the enterocyte. For fructose, it's GLUT5, which is facilitated, so it is passive by nature. And then all of them actually go into the blood via GLUT2, which is also facilitated diffusion. But for more detail, just listen a little bit earlier.
perfect. Thank you.
Yep, absolutely. Any formatting questions or anything like that?
Are there any extra credit questions that we haven't gone over?
You mean like, are there any extra credit questions that we haven't gone over in this particular Zoom session?
Yes.
I haven't gone over any extra credit questions.
Oh, why don't we go over a few of those?
I would just say that your extra credit questions are going to be coming from the fat chapter and from the video analysis too. That is as much information as I'll give you.
Okay, thank you.
Mhm. They're multiple choice this time too, so that should give you an opportunity to at least have a 1 in 4 chance.
Yes, that sounds awesome.
Dr. Maychek, can you cover the structural differences between saturated mono and polyunsaturated fats?
Yes. So the main thing that I need you to know is that saturated fats are fully saturated, so they're full of hydrogen. So if you look at the chain, all fatty acids have a carboxylic acid end and a methyl end. That methyl end is also called the omega end. A saturated fat has hydrogens on either side of the full carbon chain itself. So there are no double bonds. With a monounsaturated fat and all other polyunsaturated fats, we have specific amounts or just generally we have double bonds that would dictate the naming schemes for them. So a monounsaturated fat has, as the name would imply, one double bond. And then a polyunsaturated fat is anything more than one. So it could be two, it could be five, it could be 1,000. It doesn't really matter. It's kind of just the naming scheme there. For structural differences, when it comes to omega 3s and omega 6s, it's in reference to that methyl end, which is also called the omega end. And specifically, it's where the double bond starts relative to that end. So if you actually count the carbons, if you go one, two, three, and then there's a double bond on that third one between the third and the fourth, that would be an omega 3. And then if there is no double bond until the sixth carbon, that's an omega 6. It doesn't matter where any of the other double bonds are. So like, let's say we had a double bond on carbon 3 and on carbon 6 in the same chain, it would still be an omega 3 because it's where that double bond starts relative to the methyl end. I guess I should say, stay consistent and say it's the omega end. And the only other thing you might want to know is a trans fat. And that's literally how the orientation of the hydrogens are relative to that double bond. So if we have a double bond and the carbons are on the same side, either the top or the bottom, that would be a cis fatty acid. And a trans fatty acid is when if we look at the double bond, there's going to be one on one side and one on the other. So that would be a trans fatty acid. Another thing you might want to know is the primary states of matter for most of these fats. If you take an isolated form of saturated fat like butter, it's solid at room temperature. If you take an isolated monounsaturated or polyunsaturated fat, monounsaturated fat would be something like olive oil, it's liquid. Same thing with most polyunsaturated fats, they tend to be liquids like fish oil. It doesn't necessarily account for when it's in a food, right? So if we have monounsaturated fats like olives, olives are obviously not liquid; they are solid. And something like salmon is going to have the fat incorporated into its actual adipose tissue, so that fish aren't liquid; they swim in it. So hopefully that answered the question and also provided a little bit more detail.
Yeah, it was perfect. Thank you.
Of course, perfect. That's what I strive for. Anyone else? We got time for a few more. You got 6 minutes.
So I'm assuming there's going to be a question then where we're looking at a fat chain and we have to say if it's omega 3 or omega 6.
Yeah, you might have been absent that day, but when we went over it, I literally said, look at this, look at these questions. I did like a little test for everyone. I was like, hey, what type of fatty acid is this? And it was a picture of a carbon chain. And I literally said, I will copy and paste this into your exam. So I would look into the fat chapter, and there's some like comprehension quiz questions on there for you to be able to identify. If I asked you which of the following is a saturated fatty acid, you should be able to look at that and identify it.
Can you go over the naturally occurring sweeteners and artificially occurring?
Yeah, absolutely. The naturally occurring ones, if we're talking about non-nutritive sweeteners or NNS, the naturally occurring ones are monk fruit extract. Also, I'm going to butcher the name Lo Huan Guo, and stevia. Those are the two natural ones because they actually come from very particular, plant sources. And then the artificial ones are going to be aspartame, sucralose, ACE-K, which is the abbreviated form of acesulfame potassium. There's other ones that I'm probably not going to mention, well, there's saccharin too, that's a common one, but there's other ones like neotame, which I won't mention. I think what tripped a lot of people up during the quiz recently was they saw sucralose and they thought it was the same thing as sucrose, which is not true. It's actually in my mind, it was intentional for them to call it sucralose because it makes it sound more natural, like something they know. And from a structural component, as an aside, it is literally a sucrose molecule. They've just removed different portions of it and inserted chlorine molecules. But there was another thing in there called erythritol, which is a sugar alcohol. Those are not actually non-nutritive sweeteners. Sugar alcohols are not non-nutritive sweeteners. When you say non-nutritive, it refers to calorie content. So generally speaking, anything that's a non-nutritive sweetener, stevia, monk fruit, aspartame, sucralose, they don't have meaningful calories in them. Whereas something like a sugar alcohol, they all end with tol, maltitol, sorbitol, erythritol. They're all low calorie, but they still contain calories in them. So that's why we wouldn't consider them to be non-nutritive sweeteners. So I hope that answered that question.
So if we get a question on that video that we had to watch where it was going over all the non-nutritive sweeteners and how they were discovered, is it okay for the short answer if we were to put the scientists were like they ended up having something else on their fingers, like while they were testing different things, and when they tasted their fingers, that's how they discovered it?
Sure, if you want to relieve stress on yourself, it's not going to be a short answer. It's going to be a multiple choice question.
Oh, okay,
Yeah.
that makes it a lot easier.
Yeah, actually, since you're here, it's actually your first question. It's number one.
Oh. Well, I'm glad I remembered to ask because I was sitting here for the last 5 minutes going, there was something really important I wanted to ask him. I can't remember what it was. So,
Well, you got 2 minutes to think of it.
no, that was it.
Oh, that was it. Okay.
Yeah.
Oh, glad you remembered it. I go entire days without actually remembering what it was that I needed to remember. Any more last minute questions? Same concept for it's like all other things considered. Skip the written portions on your Scantron. Everything that's a multiple choice and true false, you answer on your Scantron. So anyone else? Bueller, Bueller, Bueller.
Just to clarify, we went over lipoproteins, right?
We did.
Okay, so I'll just rewatch it and make sure I write everything down.
Yep. If you have a hard time finding it, it was probably like a little bit more than a little bit more, a little bit less than halfway. So around like the halfway point, I believe.
Oh, okay, perfect. Thank you.
Yeah. That's my assumption. I don't have the greatest perception of time, as y'all are probably aware. Okay. I'm gonna stop the recording and I'll have this up probably in the next hour. So it just takes a little bit of time for everything to actually end up uploaded. Why do I just stop recording? Oh, there it is.